Brilliant. Thanks, Louisa. So it is my great pleasure to introduce Pia today to talk to you. So she's recently won the prestigious fellowship for R&D Lawrence Fellowship from Diabetes UK, which will be sponsoring her research for the next 5 years. So it's a great pleasure for you to come here and talk to us today, Pia. So I'll hand it over to you without any further ado.
Thank you. It's a real privilege to be here. It's become quite a tenet of what I want to do quite a lot of during my fellowship. Mostly because of the response that I've had when I've gone to days where we've had people coming into the lab or explain the biology of what I have seen in the pancreas, giving that context to what people who are living with type 1 diabetes experience, but from a different viewpoint. So I'll just share my screen, my very, very proud first screen. Can you see the screen? What have you got? Have you got the slide or the?
We got the slide on full presentation mode.
Fab. So thanks in no small part to our diabetes PPI group, I have indeed recently been awarded the R.D. Lawrence Fellowship. I think I took my original proposal to them twice, and their support and their challenges and their questions really helped me think about the project more deeply, more holistically, and more about how I wanted that to impact what people's lived experience is. So that's really the kind of slightly grandiose title, is that one researcher's perspective on how knowledge is power and collaboration is key in the battle to conquer type 1 diabetes. So I also live with a chronic condition, and I know that the first time that a clinician said to me, you're just going to have to live with it. You can't fix that, you can't fix the other, you just have to learn to live with it, gave me, it was a hard thing to hear, but it was an incredibly empowering thing to hear, because it meant that I understood my own biology. And so I think from that viewpoint, really being able to share what we as basic scientists do with people living with the conditions that we study is something that we should do a lot. But I also think that the perspectives that we get back from the people who are have the lived experience is critical to the kind of honing of our questions. And I think that together, and I think that's the way the research community is moving very, very fast and very strongly, is that huge weight of responsibility of that conversation. And I welcome it. It has been so enriching for me. So type 1 diabetes is an autoimmune condition. That means that our own immune system starts to recognize proteins in our own bodies as foreign and attack them. And in the case of type 1 diabetes, that is proteins that normally reside within beta cells. So that means that our own immune system targets beta cells and only beta cells to destroy them. And it only happens to those who are born with a strong genetic predisposition. However, having a strong genetic predisposition doesn't necessarily mean that you are going to go on and get type 1 diabetes. So it is not a test that we can do before time. We know that you are very unlikely to, or we know that you may. And so understanding more in a more refined and nuanced way people's risk of type 1 diabetes will really help if we ever want to move to a time where we can cure or prevent it. At the moment, the only treatment for type 1 diabetes is insulin. And insulin was a game changer. Before 1922, when insulin was first given to the first patient, a diagnosis of type 1 diabetes was a death sentence. However, 100 years on, type 1 diabetes, insulin remains the only therapy for the condition. We've really improved in the way we deliver that insulin, but just to put it into context for those of us who are here who don't have type 1 diabetes, a person who is living with type 1 diabetes before the closed loop system would have to give themselves around 65,000 insulin injections in their lifetime. they would have to do 80,000 finger prick tests to check their blood glucose. That is a huge burden to place. And even though there are the closed loop systems, people who are living with the condition never turn off from it. They are always having to input the amount they eat, think about the amount they exercise, what happened yesterday, what's going to happen tomorrow. It is an incredibly life-consuming condition. And so, given that incidence is also increasing by 4% each year, we know that there are environmental factors that are driving this increase in numbers. And with 1 in under 170 of us living with type 1 diabetes, we really need to get to a point where we can take ourselves beyond insulin as a treatment or a prevention for the condition. Type 1 diabetes causes an increased risk of cardiovascular disease, kidney failure, blindness, hearing loss, increased peripheral nerve and microvascular damage, and carries a huge burden of diabetes stress. Last time I was at a talk, I went after a lady who very bravely stood up and talked about the loss of her son because he couldn't manage his condition and he wasn't supported well enough by the disparate teams because of the lack of communication. We need to move forward from this. And I'm deeply committed to doing that with you. However, we still lack some fundamental knowledge. We don't know what triggers type 1 diabetes. That beta cell demise begins long before we have any indication someone is going to go on to develop type 1 diabetes. And not knowing when that beta cell destruction begins means we don't know what those triggers are. It also means that these two factors combined at the top combine to say we don't have a definitive test to predict who will be diagnosed with 100% accuracy in enough time to protect enough beta cells to prevent the disease from starting. And we still don't really understand why over a third of people who are living with type 1 diabetes find it so hard to manage their blood glucose, even when they're doing all the right things. And we don't have a therapy that modifies the conditions in all people. But today I'm going to talk to you about my case study, my work in the pancreas around these talking points. So the pancreas itself sits deep within our abdomen. It is critical for both the digestion of our food and then the balancing of that nutritional load and that blood glucose homeostasis. If you have too low blood glucose, your brain is starved of glucose. The brain requires about 50% of our glucose intake just to function normally. Too high blood glucose, and we start damaging the small microvessels and nerves. However, it sits deeply within our abdomen, and it also houses all the digestive enzymes, which if we then go to sample the pancreas so that we can study it, we run the risk of causing massive abdominal burns. So all the work that we do in the pancreas is in post-mortem or donor pancreas, of which there is only around 200 cases in the world to study the disease at onset. That is when the immune system is at its most aggressive. Going further away from diagnosis, when the beta cells are all destroyed, obviously you can't then study the pathology in action. So this is a section of pancreas. It is about 4 microns thick, which, to put it into context, is about a third of a cell thick. The blue color here is that exocrine tissue. So you can see it makes up about the endocrine tissue, which makes up about 99% of the pancreatic mass. So that's the part that creates those digestive enzymes. Dotted amongst them in a really kind of haphazard way are these tiny little islets. You can get about 10 of them on the tip of a pinhead, and there are about 1 million of them in your pancreas. They're tiny little micro-organs, each one acting independently to balance that blood glucose. So if they sense there's high blood glucose, they secrete these little brown cells here, which are beta cells, secrete insulin. if they can sense that there's low blood glucose, these little red cells here secrete glucagon. And these are the two main hormones that balance each other out. There are actually 5 types of cells within these islets that all work together to kind of balance that seesaw of insulin and glucagon. But I'll come a little bit more to those later on. So in a healthy individual, beta cell mass stays fairly steady through a lifetime. Your islets are formed really by the time you are a toddler. They are functioning, each one independently looking after you, and the cells are long-lived, and so your beta cell mass is pretty much maintained. However, in those genetically susceptible people, we think that precipitating events, and we don't know what they are. Some groups consider that it might be a virus or a bacterial infection. Some people think it's dietary in early childhood. Some people think it's a diminishment of the amount of available vitamin D, which is an incredibly important vitamin for our energy and our cellular processes. But whatever that precipitating event is, triggers our immune cells to very rapidly destroy our beta cells. This was a model that was first built by a researcher called George Eisenbarth decades ago. And the model said that when a patient reached only having 10 to 20% of their functional beta cell mass, they would go on to develop type 1 diabetes. This is mediated by a rogue army of immune cells. It's not just one immune cell. And these are the key immune cells for protecting us. So you've got CD4 T helper cells, which basically act as the generals. They kind of tell all the other immune cells what they need to do. Specifically, they tell the CD8 cytotoxic T cells, who are very efficient. They're like the snipers in the army. A general tells them what to kill and where to kill it, and these cells will do their job brilliantly. Then you've got the CD20 positive B lymphocytes. Now, these guys are the guys that make the autoantibodies that are used in detecting if a person is likely to develop type 1 diabetes within the next few months. They're also, you will have heard about, they are very important in fighting all sorts of different, many diseases. And these guys, the B cells are like special forces. They can be killers, but they can also be protectors, and they can also be spies. So pulling out the information from the local environment very effectively to tell the generals what needs killing. And then you have the macrophages, and these guys are also spies, but they also come and they do a lot of tidying up and they do some killing, but it's not very specific. And all of these are involved in that inflammatory destruction of the beta cells within the islet in type 1 diabetes. So as those people who are living with the condition will tell you wholeheartedly, it is a complex and heterogeneous disease. One person who's living with type 1 diabetes does not experience it the same as someone else living with type 1 diabetes. And some of the research that I was doing during my PhD and have done since may help us understand why. So coming back to those snipers and the special forces cells, those B lymphocytes, what I noticed during my PhD is that the profile of the immune response between individuals looked very different. Some people had really high numbers of both CD8 cytotoxic T cells and really high numbers of B lymphocytes, whilst others had very few cytotoxic T cells and almost no B lymphocytes at all. These latter individuals also retained high numbers of beta cells in their pancreas at diagnosis. Now remember that the samples that I am looking in are post-mortem, and they all died of DKA. So they all died with an absolute requirement for exogenous insulin provision. So I'll just highlight that again. You've got some patients with high numbers of immune cells, which leads to quite profound beta cell loss. And interestingly, all of them seem to be diagnosed younger. In fact, in samples that came from anyone under the age of 7, all of those individuals had this high inflammatory profile. Then you've got those CD20 low individuals where there's low-grade inflammation, really high numbers of beta cells still in the pancreas, and bearing in mind that you do get individuals who have high numbers of beta cells 50 to 60 years after their diagnosis, and these tend to be diagnosed older. And in fact, with this low profile, all the individuals we have looked at over the age of 13 exhibit this CD20 low kind of profile of inflammation. So this was really the first evidence of really distinct and stratifiable differences between individuals, and it was really closely associated with age. However, the middle group of the 7 to 12 also ascribed to one or other of those different immune profiles. So we don't think it's a sliding of a maturing of the immune system. We think there are distinct immune processes happening. But I got really curious about those beta cells that seemed to be there, but not working. So I went back to the same samples and I had a look at the insulin profiles within the samples. This here is an example of several beta cells from a healthy individual. The staining in the red is the proinsulin, which is the pro-hormone. So all hormones are made in a pro-hormone form. This is because hormones are so potent that if you make them and they're not regulated, they can do quite profound damage to sort of homeostasis within the body. So you've got the pro-hormone, proinsulin in the red, and this is localized around the nuclei, which is in an area of the cell that we can think of as the factory, the protein factory of the cell. And then out towards the periphery, we can see this green stain, which is the mature insulin. And these are really clearly separated in healthy control beta cells. And this mature insulin is now docked and ready to be called into action and secreted. So just in the schematics, you've got the proinsulin molecule is made, then an enzyme, when the conditions are right, cleaves the proinsulin into a molecule of C-peptide and a molecule of mature insulin. And indeed, when I looked at my patients, and I initially looked at these samples blinded, so I wasn't skewed by, trying to tie them in with my immune phenotype, even though that I was looking to see if there was a similarity. Half of my samples looked exactly like they did in the controls, with these really separated proinsulin and insulin areas within the cells. However, there were other patients where the proinsulin was clearly being improperly cleaved, and so was being retained within those mature insulin granules, and then we imagined they were being co-secreted. We then collaborated with our clinical colleagues here at Exeter, and we found that we could see evidence of the proinsulin being secreted alongside the mature insulin. Although there was less C-peptide, so in effect less insulin being produced in these individuals. And I gave myself away because that clicked on first. These were all CD20 high individuals, all people diagnosed under the age of 7. Whereas those with this kind of more normal looking islet were CD20 low individuals. So despite having a huge, sometimes remaining beta cell mass, they had very, very little insulin. So this led me to propose 2 different profiles. So this is the one I showed you earlier, where you have a CD high aggressive immune mediated destruction with very little beta cell mass by the time of diagnosis. But with this new yellow line here denoting the CD20 low mediated destruction, with a later age of onset and retaining much higher beta cell mass at diagnosis and beyond, suggestive of dysfunctional and possibly treatable beta cell mass. It's also important to think about the type of immunotherapies we want to subject people to, because an immunotherapy is no small is no small ask. You give someone an immunotherapy and they can no longer fight other infections as effectively. So you don't want to be giving immunotherapy to the wrong people. And if you are giving drugs that target the B cells to these individuals who are over 13 who have no B cells, obviously those trials are not going to be as effective as in those young individuals. That said, you don't want to deplete the young individual's immune response. So we've got a way to go for that, but I'll talk a little bit more about that in a moment. So going back to those differences in the immune response and going back to the differences in the behavior of the beta cell processing, those two discrete different biological mechanisms led us to propose that actual fact what we were seeing was discrete endotypes of type 1 diabetes. So an endotype is where there is a potentially therapeutic benefit of thinking about a condition as having two distinct subtypes. And as I've just alluded to, this matters because if you've got different immune targets, if you've got different causes of beta cell loss, if you have people who have beta cells that you can still target and maybe reawaken, and differences in what drives that loss of beta cell function, you may begin to understand what that kind of disparate responses between people and how they manage their condition, what trials to enroll people in, and what therapies they will ultimately need. So this isn't a family, this is a collection of individuals who all have type 1 diabetes, but may be exhibiting different phenotypes of the disease. We can now look and maybe reach a time where we can put the right people in the right clinical trials. But not only in the immunotherapy trials, we need to incorporate this understanding into all the areas that we are researching. Are there differences in the causes and the triggers and the way we diagnose individuals according to their endotype? What about how we manage them and how we help people understand the need to treat their diabetes? I've had several people come to me and say, you know, maybe it's that I've got more beta cells in my pancreas, and that is why I have these days where suddenly I've got far too much insulin, even though I'm doing exactly the same thing as I did yesterday and the day before. And maybe by incorporating all of this knowledge, we can reach a time where we're talking really realistically about cure and prevention. To do that, we need to keep working. And we're at a time now where we have really advanced technology. So whereas during my PhD, I was counting immune cells in ones, literally, I had a pen that I was dotting 1, 2, 3 in units, how many B cells, how many T cells. I can now use a piece of equipment we have here to look at a whole pancreas section. That's 1 million cells. And I can look at 8 proteins within that to really understand the nuanced differences between different beta cells. I can look at whether they are old beta cells, healthy beta cells, stressed beta cells, in the context of which immune cells are around them. And this gives us a really powerful way of really interrogating this really rare tissue. In fact, my fellowship is going to be asking that kind of nuanced question about how these beta cells can respond to glucose, how they may respond to protein proinsulin synthesis and processing, trafficking and storage. Are there proteins missing from that glucose response pathway in the TIDE2 islets that are still there 50 years after diagnosis? What is impacting glucose secretion and beta cell survival? And are there different beta cell subtypes that if we lose some of them, we lose all of the islet function? And tantalizingly, in people who have had TIDE2 for 5 years, I see evidence of new beta cells. Now, does that mean that beta cells are forming in all people, but the immune system is killing them off? Or is there something special about those TIDE2 pancreas that we could then harness to really help replenish that loss of beta cells? In between the lines, we can really ask nuanced questions. And I can't do all of this in the pancreas, but I can do it in collaboration with mathematicians, with cell biologists, with mouse biologists, with geneticists, with clinicians. So how we beat type 1 diabetes and many, many other conditions, I really believe, is using our huge technological advances, coming out of our silos, really working together, finding ways to get around the funding trap where we need to keep our own science to ourselves so that we get funded on the next round, finding a way that we can share equitably our data and utilize it in a really holistic and integrative manner. Because then what we can start doing is really tying together all the different types of research and feeding backwards and forwards between each other so that we can really speed up our discovery profiles. This has already led to improvements in beta cell replacement therapies. So whilst the pancreas kidney transplant is still the gold standard for people who really, really struggle to manage their blood glucose, despite doing all the right things, obviously the donor pancreas and kidney is rare, but it carries a huge burden for patients. So we have now improved transplants for donor islets, but even these still only last 10 years and still require immunotherapy, which carries a burden, as I described previously, for people in receipt of transplanted material from another individual. We are really working hard to develop stem cell-derived beta cells. So this is cells that are converted from one type of cell, from a pluripotent cell, which has a fate to be able to be turned into any type of cell in the right conditions, to be able to generate as many beta cells in the lab as we want to, as we need to. But again, we need to make sure that these cells are allogenic so that they don't trigger our own response to kill the alien cells. We have got really, really hopeful immunotherapies on the horizon. So we have a drug called rituximab, which has been shown to slow down the loss of endogenous insulin production, but only for a few years and not in all people, which makes sense when you think back to the story I was just telling about how some people don't even have B cells within that inflammatory attack of the beta cells. There are other drugs that target each area of the immune system, and the teplizumab study is causing huge excitement because actually it's been shown to delay the diagnosis of type 1 diabetes for 2 years. And this is incredibly important for people's health and long-term well-being and long-term management of their condition, and should be licensed within the next couple of years. However, what these sort of knocking out these whole swathes of the immune system still leaves you really vulnerable to infection. And so increasingly, we are looking at technologies that allow us to target only the B cells or only the T cells that specifically target the proteins that are driving forward that autoimmune, that auto attack on our own cells. There are a host of intervention studies that are being carried out. As I just described, there's islet transplantation and stem cell therapies being developed. There are also preventative studies. These two studies here in the purple are studies that believe that viruses, which have been shown to have a huge correlate with the onset of type 1 diabetes, are being undertaken to see whether that can prevent the onset. There are interventions around diet, and there are interventions around managing the condition and repurposing drugs that have previously been proven safe, but maybe not used in this context, and new types of drugs that will help us move forward to being able to really protect ourselves from that loss of beta cells. So if you combine all of those things together, we really are moving to a time where we have precision medicine, and we will understand, in this case, type 1 diabetes, so in such a nuanced way that we will understand which people require which therapies or which people should go into which clinical trials. So where we're coming from to where we are going. Whilst we still don't know the triggers for type 1 diabetes, there are increasing longitudinal studies and prevention studies and vaccine trials that may give us the answers. And although we still don't know exactly when beta cell destruction begins, there are increasingly refined methods for testing serum to see when you can start detecting beta cell damage in the serum. And although we don't have that definitive test to predict who will be diagnosed with complete accuracy. Work being carried out here in Exeter has already advanced this aim with the genetic risk score that was developed by Richard Oram and his colleagues, and is now being extended to bring in other markers to make that even more predictive of type 1 diabetes. And as we understand more, we will be able to tease apart which type of type 1 diabetes you have. And by understanding the biology in the pancreas, it also helps us to really think about how we manage it and how we convey the information that we need to clinicians and to people who live with the condition. I find it incredibly moving when I sit in front of the microscope with some visitors and we talk through what is happening, what the immune cells are. It's an incredibly enriching experience, and I think to carry that further and more deeply will also speed up all our findings. So whilst we might not be there, I think we are getting to the beginning of being there, which is really exciting. Things to consider is that, as I've kind of gone on through this entire presentation, that integrating that lived experience and that basic research, I think, is incredibly important. I think that we also need to consider information that we maybe garnered a long time ago, but then didn't know its importance because we didn't know that maybe we were looking at different disease types. So maybe clinical trials that didn't quite work maybe need to be revisited in the context of putting the right people in the right trials. It's also really important that we share the data we know. And this is a big discussion that is happening right across the basic science communities, is how we have open access data. But we also have to be able to share our data in ways that is easily understood. So it's all very well sharing really complex data sets, but if no one knows how to access them or interrogate them, there's really no point in sharing them. So that is another conversation that I think you will see coming through more and more in grant applications, is on how they're going to share data and how they're going to make it accessible. More and more samples, always more samples, which is such a huge ask for the diabetes or the volunteer communities, but they really are fantastically important and they are utilized really well and they really help. And then also that idea that, so a long time ago, we did lots and lots of studies in mice. We got very excited about the findings. And then a few studies showed that actually we may have gone down the wrong track because we were looking at mouse biology and not human biology. And suddenly it was like, no, we don't want to do any mouse biology anymore because it's not human. But we can't do the studies in humans that we can do in mouse or model systems. So it's that careful integration of both, that they feed each other accurate knowledge within the limitations of their remit to really build knowledge quickly. But it has, I really believe that there is a strong place for both. And yeah, embracing that new technology and the computing capabilities. I got told that I was too old, I was never going to learn coding. I started learning art. It's very hard, it's very exciting. But I think that being able to really use data for all of us is something we have to do, or we have to at least acknowledge that we need help with it so that we can appropriately factor it into our grant applications. But I do think that working together, we really do have a really great shot at moving everything forward much faster than we have so far. And that's going to be all of us working together. In my case, I'm lucky enough to work with Diabetes UK, who are a phenomenal charity. I went to their collaborators, their colleagues' day, and a more dedicated team of people you could not hope to meet. But also, I'm really lucky that I work at Exeter. I work in an extraordinary team with extraordinary mentors and colleagues and collaborators and the technical services team and the professional services team. You know, together we make things happen. And I think it's really important that we continue to celebrate that togetherness as we push forward. Thank you. That was very passionate. I'm really sorry.
Please never apologise for passion. It makes the presentation so good.
Right.
Thanks, Pia. That's brilliant. Are you able to stop sharing a second?
Yeah, I'm trying to work out how I do that. How do I do that? Where do I do that? How can... Oh, there.
It should be top of the screen, I think.
Yeah, there we go. Yeah, sorry, it had disappeared off the top and my screen is so large. Hello.
But
I'm
thanks for that.
I'm really happy to take questions, but I'm also really happy to be emailed with any questions that you might sort of think about afterwards about this presentation, or if you read anything, or, and I can't promise I will know the answer. I am an early career researcher, but I probably will know somebody who will, and I'm really happy. It helps me develop, but I'm really happy to go and find someone who does and get back to you with anything that, you know, strikes you or you're curious about.
Thanks, Pia. That's great.
Share your email address then, Pia.
Yeah, I will. I
Okay,
will.
thank
I'm
you.
really happy to do that.
Is there any questions for Pia a moment? Bob.
I hadn't appreciated how difficult it is to actually work with the pancreas in vivo. Is there any scope for remote sensing technology such as MRI?
So there are MRI studies being done, and there are interesting results coming out that show that people's pancreas tend to be smaller if you have type 1 diabetes. It tends to be smaller in those people who develop autoantibodies. So that might be a way of looking to see whether somebody who has autoantibodies may go on to develop type 1 diabetes. But if you think about the pictures that I showed you, that is you're looking at nanometers, and to use that kind of technology to look at that level, we can't yet do. We can do it in mice, but again, not at the confocal level. But there's a lot less mice to kind of get through with ultrasound. But even then, you know, you're not able to see at a cellular level.
Okay, thank you.
Okay, there's lots of hands up and things. I've just got a list, so going in the order I noticed them. So the next one was Alexis. I saw your hand pop up just now.
Okay, right, thank you. I'm unmuted, am I? I can't
No,
find
yeah,
I can hear you. Yeah.
Okay, it's really good. There were sort of two threads I wanted to follow. The first one to say was thank you very much. That was a brilliant presentation. I love the way you had your metaphors as well. It was really good. Can't profess to understand more than about 1% of it and want to read it again and think about it again. But thank you very much for it, and indeed for including us. I've got two sort of strands of thoughts really, and I don't know which way to go. I'll go the short way first, which is the beta cell. Do you know a lot about it? Have there been vast studies about it, the physiology, the anatomy, you know, everything about it? Or is it actually a bit of a blank?
No, there have been enormous studies about the beta cell. So okay. Not by me, but there are studies about, it has been studied deeply for many, many years. So about the electrophysiology of it, about the enzyme processing within it, the protein production. Increasingly, we know more and more about how the beta cells themselves protect themselves against inflammation or stress. There are huge studies about beta cell stress and the proteins that are made when they are stressed, because obviously a beta cell has to work very, very hard to make those insulin granules. You know, there are millions of insulin granules in each beta cell, and that level has to be maintained. And so that's a lot of hard work. So yeah, there are massive studies, but I have to be honest, a lot of the studies are mind-blowingly complicated. So when you get into the millivoltage of how an insulin granule or an insulin vesicle binds to the cell membrane so that it can secrete that insulin, it's like, so we each have, so yeah, there are enormous numbers of scientists looking at every single aspect of a beta cell to understand it.
Excellent. I can see some sort of models being made, and you go to a conference and there's a huge model of a beta cell and it's dissected.
Oh,
you know,
can
it must
we
be
make
brilliant.
one together?
Yes, that would be brilliant. I think you need smartest. That's coming on to my second thing, just very quickly, because I mustn't hog everything. In a field in which I work, which is nothing, or have worked rather, nothing to do with science really, we think about working together. Somebody termed it, and I can't remember their name, the worst contemporary issue, and they called it a wicked issue, trying to work together across disciplines, across sciences, across, you know, the whole lot. It's really, really hard. And I think we've got better over the time that I've been alive, but it's still a struggle. And I just wondered whether you had anybody who was outside, but was outside of your group. You showed a picture of it, whose job was to think about how well does this group actually function together, apart from over the water, you know, the coffee or whatever. Really, really, in terms of nailing things down, talking to each other, the whole structure of how you work together. And it's not only you in that photograph, it's all the world, isn't it? And those things, I think, need people whose jobs is to maintain a very close look and a very distant look at how this is functioning, where the gaps are, and how they need to be, you know, pushed maybe with some soldiers or some snipers or some whatevers to get over hurdles, the silos, which are so resistive. Anyway.
They are so resistive, but I really, really believe that we are moving away from them. I believe that collectively we are, and we're not just scientists talking to scientists or scientists talking to clinicians. I've had a collaboration with an artist, and she made this beautiful installation of hundreds of these beautifully painted slides that she'd made out of one of my confocal images. And each slide had a story written by somebody with type 1 diabetes on the little slide holder. So, you know, there is that kind of conversation happening, not only between scientists and people who are living with the conditions, but also out to the wider public. And in terms of multidisciplinarity, that is, at the moment, it is the key word. Everyone's talking about it. Everybody's talking about how you bring together different thought processes, different priorities. You know, I've got a collaboration going on with a mathematician, and it is happening, but that is where you guys are key. You keep us doing it. You know, conversations with coming to the PPI. I mean, that first time I brought my talk, I was terrified, and everyone was so kind. But they absolutely, you know, I was told it, you know, it doesn't really make that much sense to us. And that made me think about, okay, so what am I doing? I'm not just doing some random protein that's just interesting to me. This actually has context. And once it has context, that's when I realized that that's what I wanted to do with that whole study around the beta cell function. So I think, you know, that I think that is where we are.
Brilliant. Annette. Sorry.
I'm just wondering, Pia, thanks for a really brilliant talk, lovely clear slides, and, you know, pitched at a level that most of us could get most of it, understand most of it. I'm just wondering, given the sort of exponential rise in the number of people with type 2 diabetes, whether there's any potential overflow from research from type 2 to type 1, and indeed vice versa. Type 2 is usually associated in the public mind with high body mass index and sloppy lifestyle choices and what have you. And there's a huge burden of stigma. And I'm wondering if there's anything that can be learned from type 1 that might actually, just as you're trying to classify different types of type 1, I know there's some classifications of different types of type 2, but whether we can break that down further and have more targeted, individualized therapies and advice for people with type 1. So I would like to see more overflow or between the two, or evidence of, you know.
So that does happen. That does happen, particularly, I mean, there's been a lot of work done here at Exeter to tease apart those people who are being diagnosed with type 1 diabetes after their 20s. You you know, because immediately people think about type 1 as the young one, the one that happens in teenagers or children. And so people were being misdiagnosed as having type 2 when they were type 1. And then there is the emerging conversation about LADA, which is latent autoimmune, mature onset of type 1 diabetes. And again, it's that, and there are genes that are associated within that remit. There are genes that are associated both with type 1 diabetes and type 2 diabetes. So I think there are crossovers. I don't think people ever act in isolation, just thinking about their own condition. But I think as well, we do need to stay focused. But I agree that I think that removing stigma by understanding the biology, and I think the sloppy lifestyle idea, you know, people make choices for a million reasons and their bodies respond in a million different ways. I cannot eat carbohydrates, you know, they hurt, but there's no way of knowing why that is. So for somebody who needs to eat differently, I think, you know, these conversations are happening. I agree that there needs to be a reduction in stigma, but I think we've probably got a long way to go, but people are looking at both in the context of each other to see what can be learned.
There's also been a group in Sweden that have essentially trying to do what they call cluster analysis and trying to find subgroups of people who have diabetes and say whether some of it's more metabolic or some's more genetics. And they're not kind of defining it in type 1 or type 2 as such. They're just kind of trying to put the data and pull out what the different groups are to see what that can tell us and whether that can help go down the personalized medicine route. So I think there's a lot going on. There's just not a clear one story coming out so far.
And so I think that's partly because we're coming from different angles, aren't we? I think that study is looking at people's responses almost first. So actually, rather than giving the label, they're looking at what benefit do they get from particular therapies and clustering them that way. Because although my argument is you start in the pancreas right at the beginning, actually, if you can get to where you want to be at the end of the story without having to climb that mountain, then how great would that be? So I think you know, there's an argument for keeping going with it all, and eventually it will all come together if it's the right story.
Yeah, I think it's just there's more going on than we realise, I think is the message. Yeah.
Thanks, Pia. There's a lot of other questions lining up here now. Thank you very much.
So, Norina, what?
Well, thank you. Mine are some simple numbers questions, please. How many people nationally have got diabetes type 1? The quality of life, as you've explained, is severely impacted, but is life expectancy noticeably shorter with sufferers from type 1 diabetes? And the 4% increase you talked about, is that real, or is it better diagnosis earlier on? From adverts I see on the telly, I would think that monitoring is getting better. We're out there constantly seeing adverts for blood sugar indicators that you can get these days. So, yeah, on the numbers side, I'm interested in. Thank you.
So the latest figures that I got for this talk is that 1 in 178 of us are living with type 1 diabetes in the UK. And that's, I mean, that was staggering for me. That was a staggering number. In terms of better diagnosis, yes, I think that might cover some of that, because we are diagnosing people better and we are diagnosing people later. So people that were being shunted into type 2 just by the fact of their age are now being classified as type 1 diabetes. But if you think of the number of people with type 1 diabetes and you think of 4% of that amount of this population, I think it can't just be better diagnosis. That's a lot more people in a population as large as the UK. In terms of life expectancy, yes, sadly, type 1 diabetes still impacts life expectancy, and it still impacts life expectancy quite considerably. I think the numbers are around 17 years on average. But what is also striking is that when you then tease that apart into the different age groups, if you are diagnosed under the age of 10, that becomes a greater than 20-year expected life loss. And when you also then think about, I spend a lot of time, not so much personally, not so much thinking about life expectancy, but thinking about healthy life years, because really those are the life years we want. I think that the technology, as you say, is fantastic. I think it is life-changing. You read about, you know, the impact that having being on a closed loop system can have for people. It is enormous, and the work that Partha Kar has been doing to try and get that pushed forward so that it is available for everyone is, I mean, no words really. But yeah, I think we have a way to go. But I think there were a lot of questions there. Did I get all of them?
Yes, I just wanted to, how many people, so you've answered that. You confirmed the quality of life and the life expectancy and whether it's real or diagnosed increase. So you're very clever even to remember the questions.
I surprised myself.
Yeah, I'm just a bit aware of time. We've still got some questions. Sorry. So, Robin.
Thank you. Yeah, Pia, once again, risk of sounding bored, brilliant presentation. Thank you very much indeed. The technology side is fascinating because you mentioned about how many finger pricks people have to do. Having a sensor on the arm now, you can test your blood however many times a day without giving money to Roche for their BM sticks, without wondering what to do with your blood bits and pieces, tissue paper. You know, just about Partha Kar. I first met Partha when he was a junior doctor at the QA at Oxford through my wife, who used to be a diabetes specialist nurse. He's really gone up in the world, has he not?
I think now that we've got closed loops, I mean, yeah, that's amazing.
It is. It absolutely is. But I mean, I think the other thing is that to me, the message of diabetes, okay, all credit to my wife, if you can really work hard at managing your control of your blood sugar, it's not so life-limiting as you might otherwise believe. A lot of the life-limiting stuff comes because people don't bother to monitor and control themselves properly. And it's still difficult, mind you, but it's not a hopeless task anyway. Thanks once again.
I think that also, I don't know, because I don't live with type 1 diabetes, but I think that there are a lot of reasons people find it hard to maintain that level of focus that are beyond just the biology. My daughter's boyfriend really struggles, but he really struggles because he came from a really tough life. He learned to manage it on his own, and you can't disassociate those choices with the rest of life. And I think coming back to that stigma thing, I mean, if we can get to a point where people are helped, where they may struggle to help themselves, I mean, we all want to do the best we can, don't we?
Yes, yeah. But the new tools are certainly helping. So thanks once again.
And Michelle.
Thank you. I promise I'll be super quick. But Pia, thank you. That was just absolutely great. And just two very quick things. One, it really resonated because I live with type 1 diabetes. I was diagnosed when I was 26. I was thinking, oh, I wonder if I'm in your number 2 group because I've been told I've got high levels of C-peptide, and
I'd lay money on it.
Yeah, so I'm sort of fascinated and intrigued about what your work might mean in practice and over what time period that might yield something actionable. I think it's really exciting, and I don't expect you to know the answer to that. But related to it was really an offer and an ask from you as to what help you need from the community in the next phase of your work from a kind of type 1 diabetes perspective, if you need more patient involvement, if you need people to get involved in trials or whatnot. So it'd be interesting to hear if there's anything we can do to help.
Trials is difficult for me because I'm not going to come and ask you for a teaspoon of your pancreas.
No.
But having you on side, maybe if you're interested and would like to read the papers as I generate them. So although they will be heavy and dense and scientific because they'll be going to scientific journals, they still need to be accessible and well written. And I think I probably have a journey there. Having you guys come to the lab, talk through what you see on the microscope. Each time I talk about it, when I'm looking at it and people ask me questions, I think about it in a whole new way. Every time, a different perspective, a different mind, looking at the pictures. You'd be really welcome, and it helps. It wouldn't be a burden to have you all visit.
Okay, brilliant. Thank you. I'll I'd love to come in. That sounds brilliant.
Well, let's be in touch. Let's try for something.
Yeah,
fab. Really great.
Thank you. That was it from me. Sorry, we've gone over.
Okay. No, it's just a bit aware that some people might have other commitments, so I don't want anybody not to have a chance to ask their questions. Is there any final questions for Pia at all? No. Okay, then I'd just like to thank you very much, Pia, for the talk. It was absolutely brilliant. So thank you very much for coming. And thank you. And it was lovely to see everybody else, all our representatives today. And I look forward to seeing you in person, a lot of you at the Lay Day in a few weeks. Won't be long.
Can I just ask, Alexis, if you have any ideas about thinking about that team dynamic, and I'd be really interested to hear about that as well, especially as I'm now going to be developing my own team and how to build a team that is strong and able to then communicate with the kind of wider world. That would also be really helpful to me.
Okay. If I can say, I've done a lot of work in team working as a consultant. I'll happily drop your email details and I'll send you some ideas for things you can work with. Very practical tools, which helps you understand the behaviour of individuals. It also helps people come out of silos as well. Okay.
That would be great, because that's also that talking to other groups.
Maybe Robin, you and I could just have a conversation, and maybe as a
Please
pair we could communicate. Sure. Louisa will get us emailed.
Yes, indeed. Okay,
that would be amazing. Be amazing.
Thanks,
thank you. Okay. Bye.
Okay.
Bye.
Take care, everyone.
Bye.